വെൽക്കം ടു വെറ്ററിനറി. ഇന്ന് നമ്മൾ നോക്കാൻ പോകുന്ന ബയോകെമിസ്ട്രിയുടെ ഫസ്റ്റ് ചാപ്റ്ററാണ്, ദാറ്റ് ഈസ് ദ സെൽ ബയോകെമിസ്ട്രി. കമ്പാരറ്റീവ്ലി ബാക്കിയുള്ള ചാപ്റ്റേഴ്സിനെ വെച്ച് നോക്കുമ്പോൾ ഈസിയുള്ള ചാപ്റ്ററാണിത്. ഇതിലങ്ങനെ എസ്സേ ടോപ്പിക്സ് ഒന്നും ചോദിച്ച് കണ്ടിട്ടില്ല. ഷോർട്ട് ആൻസേഴ്സ് ആയിട്ടും പിന്നെ കുറച്ചും കൂടി ഇമ്പോർട്ടൻ്റ് ആയിട്ടുള്ളത് ഗ്ലൂക്കോസ് ട്രാൻസ്പോർട്ടേഴ്സ് ഒക്കെയാണ് ചോദിച്ചു തന്നിട്ടുള്ളത്. സോ നമുക്ക് ചാപ്റ്ററിലേക്ക് പോകും. സോ സെൽ ബയോകെമിസ്ട്രി. First of all, what is cell or what are the components of cell? Cell contains cell membrane plus protoplasm, and this protoplasm contains cytoplasm is nucleus, and cytoplasm consists of cytosol and intracellular organelle. So നമുക്ക് ഇൻട്രാസെല്ലുലാർ ഓർഗനലാണ് ഇതിൽ മെയിൻ ആയിട്ട് ഇമ്പോർട്ടൻ്റ് ആയിട്ട് ചോദിച്ചു കണ്ടിട്ടുള്ളത്. സോ അതിലേക്ക് പോകും. ഇൻട്രാസെല്ലുലാർ ഓർഗനൽ, അതിൽ ഫസ്റ്റ് ആൻഡ് ഇമ്പോർട്ടൻ്റ് വൺ റൈബോസോം. ആൻഡ് റൈബോസോമിനെ കുറിച്ച് പറയുമ്പോൾ നമ്മൾ ഫസ്റ്റ് തന്നെ പറയേണ്ട ഒരു കാര്യമാണ്, ഇറ്റ് ഈസ് എ ന്യൂക്ലിയോപ്രോട്ടീൻ organelle. Why it is called a nucleoprotein organelle? Because it contains both rRNA and proteins, and we know that in both prokaryotes and eukaryotes, ribosome consists of two subunits. And in prokaryotes, it consists of 70S total, which contains 40S large subunit plus 30S small subunit. And in eukaryote, it contains 80S ribosome with 60S large subunit and 40S small subunit. And normally these two subunits are in dissociated form, but its association takes place during the protein synthesis. So next we move to the functions of ribosome. Ribosome is called as the factory for protein synthesis because along with the ribosome, the mRNA, tRNA interacts to synthesize proteins. So factory for protein synthesis. And ribosomes are present along with the endoplasmic reticulum to form rough endoplasmic reticulum, which helps in the formation of proteins and export these proteins out of the cell. Synthesize and export of proteins. Next, certain ribosomes are present in free form in the cytosol. These free ribosomes produce proteins which are used within the cell. So that's all about the protein. Next we move to the Golgi apparatus. So next, the Golgi apparatus. Golgi apparatus is the flattened membrane surrounded vesicles, and mainly its functions includes protein modification. Functions: first, protein modification, its packaging and secretion. Modification plus sorting is there. So what is protein modification means? In the rough endoplasmic reticulum, proteins are synthesized with the help of ribosome. From there, these proteins are carried to the Golgi apparatus, where it is modified by adding carbohydrates in order to produce glycoproteins. That is, these proteins plus carbohydrate gives glycoproteins, and proteins which get added up by lipids form lipoproteins. And this is first function. Next, it also helps in the formation of lysosome and peroxisome. So that's all about Golgi apparatus. So next we discuss about mitochondria. And mitochondria is the powerhouse of the cell, that we all know. And mitochondria illathora sthalam, almost every cell contains mitochondria, but mature erythrocytes do not contain this mitochondria. And about the structure, the mitochondria contains two membranes, outer membrane and inner membrane. Outer membrane is smooth and continuous, whereas inner membrane has certain invaginations called as crystal, and the space between the two membrane, that is inner membrane and outer membrane, called as intermembrane space. And the space inside the inner membrane is called mitochondrial matrix. And in the matrix, we contain certain circular DNA called as mitochondrial DNA, and certain 70S ribosome, RNAs, etc. So this one thing about the mitochondrial DNA is that this mitochondrial DNA is inherited. We get this mitochondrial DNA from mother. So mitochondrial DNA disorders like a mitochondrial myopathy is transmitted from mother to the fetus and not from father to the fetus. Then going for the functions of mitochondria. We know that mitochondria is the powerhouse of cell. Why? Because it produces ATP. So functions is the inner mitochondrial, that is this inner mitochondrial membrane contains certain electron transport chain. Electron transport chain inner membrane with electron transport chain and oxidative phosphorylation, and it carry out oxidative phosphorylation. So the energy released from the oxidation of food molecules, that is glucose, fatty acid, amino acid, ഇതെല്ലാം ഓക്സിഡേഷൻ ചെയ്യുമ്പോൾ we get NADH plus, FADH2. These all get converted from these intermediates or these compounds. We get ATP through this ETC. So ATP is produced in the mitochondria, and inside the mitochondria certain metabolic pathways takes place, which includes tricarboxylic acid cycle, that is TCA cycle, beta oxidation of fatty acid, pyruvate dehydrogenase reaction, ketogenesis, ketolysis, and certain parts of heme synthesis pathway, gluconeogenesis, parts of pyrimidine synthesis. These all takes place in mitochondrial matrix. So these all are the main functions of mitochondria. So next we are going to discuss about the peroxisome. So a peroxisome is a single membrane structure enclosing a granular matrix. Granular matrix ഒരു സിംഗിൾ മെംബ്രെയിൻ എൻക്ലോസ് ചെയ്ത ഈ ഒരു സ്ട്രക്ചറാണ് പെറോക്സിസോം. സോ over to the functions. Peroxisome contains two enzymes, catalase and peroxidase. So these two enzymes destroy peroxides and free radicals. So these protect cell from their toxic effect. And peroxisomes are also involved in, this is the first function. Second one, peroxisomes are involved in the synthesis of plasmalogens and glycolipids. So first one, it's protect the cell from the toxic effect of peroxides and free radicals. Second one, it helps to synthesis of plasmalogen and glycolipids. Next, third one, peroxisomes are also involved in the oxidation of long chain fatty acid. Oxidation of long chain fatty acid. And one thing in this comes under this peroxisome is the cell Weger syndrome. Cellular syndrome occurs due to the inherited absence of functional peroxisomes in all tissues. It is characterized by neurological impairment and child dies in first year of life. This occurs when there is absence of functional peroxisomes. So what happened? It cannot protect certain cells from the free radicals, so it mainly affect the neurological tissue, which cause neurological impairment and cause child death in the very first year of life. So this is all about peroxisome. So അടുത്ത നമുക്ക് ലൈസോസോമിനെ കുറിച്ച് നോക്കാം. So lysosomes are structurally an organelle which contains small vesicles surrounded by a single membrane. Small vesicles containing certain enzymes surrounded by a single membrane. And we know that a lysosome contains many enzymes, many hydrolase enzymes. So it contains an acidic pH which is approximately around 5. That is, it is pH is less than that of pH of cytosol. Next, moving to the functions. So lysosomes are responsible for degradation and recycling of intracellular biomolecules. That is, we know that lysosomes contain certain enzymes, certain hydrolytic enzymes, which are involved in the degradation of intracellular biomolecules. That is, degradation of സെല്ലിനുള്ളിൽ ആവശ്യമില്ലാത്ത ഇൻട്രാസെല്ലുലാർ ബയോമോളിക്യൂൾസിനൊക്കെ ഡീഗ്രഡേഷൻ ഹെൽപ്പ് ചെയ്യുന്നതാണ് ലൈസോസോം വിത്ത് ഇറ്റ്സ് എൻസൈംസ്. Next, second, it digests the subject brought by the cell by phagocytes. That is, it helps in the digestion of substance that enter the cell through phagocytosis. And this occurs when, if this is a cell and another particle, the cell endoside this particle, and now the particle is inside the cell, and there is lysosome inside the cell with so many enzymes in it, and this lysosome combines with this endocytic vesicle. Now this became an endocytic vesicle. So this is primary lysosome. So the primary lysosome combines with the endocytic vesicles to form a secondary lysosome. So primary lysosome plus endocytic vesicle form secondary lysosome. And this secondary lysosome digests the foreign particles. Foreign particle within it, and the products formed are utilized by the cell. This is how it's helped in the digestion of endocytic particles. Next, moving to the enzymes present in the lysosome, it contains various hydrolases, which include carbohydrate hydrolyzing enzymes like beta-galactosidase, alpha-glucosidase, alpha-fucosidase, mucopolysaccharidase, alpha-mannose, etc. Then protein hydrolyzing enzymes like cathepsin, elastase, peptidase, collagenase, etc. Next, nucleic acid hydrolyzing enzymes like ribonuclease, deoxyribonuclease, next, now it is carbohydrate hydrolyzing enzyme, protein hydrolyzing enzyme, nucleic acid hydrolyzing enzyme, and next is lipid hydrolyzing enzymes, which include fatty acid esterase, phospholipase, Next we are going to discuss about lysosomal storage disease. So we know that lysosome contains various enzymes which digest carbohydrate, protein, lipid, nucleic acid, etc. So if any of these enzymes are absent, what happens? These excess amount of these either carbohydrate or nucleic acid, lipids get accumulated in the cell, isn't it? So these lysosomal storage disorders are a group of disease that occurs due to the absence of specific lysosomal enzyme, resulting in abnormal storage of related compounds in lysosomes. Resulting what? Abnormal storage of compounds in lysosomes. There are various diseases under this category, which includes Pom's disease. It is due to type 2 glycogen accumulation. Next is Niemann-Pick disease, Tay-Sachs disease, due to accumulation of sphingolipids, that is sphingolipidosis. Next type is Hurler's disease, Hunter's disease, due to accumulation of mucopolysaccharide, that is mucopolysaccharidosis. So these are all about lysosome. First structure, then function, then lysosomal storage disease. So next we move to the endoplasmic reticulum. So endoplasmic reticulum is a membranous channel system connecting the nuclear membrane and plasma membrane. So this endoplasmic reticulum is a membranous channel that connects nucleus with the plasma membrane. It have network-like structure. Then these are two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. What is rough endoplasmic reticulum? It has a rough appearance. Why? Due to presence of ribosome. So due to the presence of ribosome, we know that it helps in protein synthesis. Next is smooth endoplasmic reticulum. It has a smooth appearance. Why? Because it has no attached ribosome. Then over to the functions. Endoplasmic functions. Endoplasmic reticulum provide a mechanical support. mechanical because it is present in the cytosol connecting the nucleus with the plasma membrane, and it is a network-like structure, so it's provide a mechanical support to the cell. And it also helps in the transport of synthetic products like glycoproteins and lipoproteins out of the cell. So second one is the transportation of synthetic products like glycoproteins and lipoproteins out of cell. Then mainly the smooth endoplasmic reticulum helps in lipid synthesis, that is lipid synthesis, which include triglyceride, cholesterol, phospholipid, synthesis. Also it helps in what? Steroid hydroxylation. Steroid hydroxylation and detoxification of various drugs. Detoxification of various drugs. And we know that a rough endoplasmic reticulum also helps in protein synthesis because they are associated with ribosomes, and it also acts as intracellular reservoir of calcium. So these are the main functions of endoplasmic reticulum, that is it provides mechanical support to the cell. It helps in the transportation of synthetic products like glycoproteins and lipoproteins out of the cell, and it helps in the lipid synthesis, which include triglyceride, cholesterol, phospholipid synthesis, and it helps in steroid hydroxylation and detoxification of various drugs. So next we going to an important topic, that is the marker enzyme. So what is marker enzyme? Marker enzyme are the enzyme that present only in one particular organelle, that is it is present only in one particular organelle. So after centrifugation, the separated organelles are identified by detection of the marker enzyme in the sample. So the use of marker enzyme is that it helps to detect the separated organelles during centrifugation. So there are various marker enzymes for various organelle or cell components. So the marker enzyme present in various organelles or cell components are In mitochondria, the marker enzyme is ATP synthase, and in lysosome, marker enzymes are cathepsin, and in peroxisome, marker enzymes are catalase, and in endoplasmic reticulum, marker enzyme is glucose 6-phosphatase, and in Golgi complex, marker enzyme is galactosyl transferase, and for cytoplasm, it is lactate dehydrogenase, and for nucleus, it is DNA polymerase. So, marker enzymes are for mitochondria, it is ATP synthase. For lysosome, it is cathepsin. For peroxisome, it is catalase. For endoplasmic reticulum, it is glucose 6-phosphatase. For Golgi complex, it is galactosyl transferase. For cytoplasm, it is lactate dehydrogenase, and for nucleus, it is DNA polymerase. So these, we have to remember these organelles with their marker enzymes. So next, we are going to discuss about one of the most important topic, that is glucose transporter. Glucose transporters are various membrane-bound carriers which helps in the transport of glucose. So we know that after digestion of starch or carbohydrate, the monosaccharide, that is the glucose, get absorbed from the lumen of intestine to the intestinal epithelium with the S-GLUT1, and this S-GLUT1 helps in the transport of glucose along with the gradient of sodium. So glucose from the lumen reaches the intestinal epithelium through S-GLUT1, then from the intestinal epithelium into the blood capillaries through the GLUT2, that is glucose transporter 2. And this glucose transporter 2, it is a facilitated transporter which helps which function in a uniport mechanism and in a pick-pock manner, that is the glucose in this transporter. For this transporter, maybe this is the transporter, and here glucose binds, and when it binds here, the first binding site get closed, then it changes the configuration, and the binding site now get exposed to the inner side. binding site now get exposed to the inner side. This is outer, this is inner. First, glucose from outside the cell bind to the first binding site. Glucose bind to the first binding site. Then the transporter undergo configuration changes and resulting in the closure of this binding site. Now this binding site get now exposed to the inner side of the cell, and the glucose is now facing the inner side of the cell. Then this get released into the cell. This is ping pong mechanism. Next, there are various types of glucose transporters, which includes GLUT1, GLUT2, GLUT3, GLUT4, GLUT5, GLUT6 is not there, GLUT7, then S GLUT1 and S GLUT2. So GLUT1 is present in RBC, kidney, brain, and GLUT2 is present in this serosal side of intestine cells. Serosal side of intestinal cells. Then liver, etc. Then GLUT3. GLUT3 is present in brain and neuron. Then GLUT4 is present in heart, skeletal muscle, adipose tissue. Then GLUT7 in liver, endoplasmic reticulum. Then next, GLUT1 in intestine. Sorry, I missed GLUT5 now. So GLUT5 in small intestine. Testis, kidney, then exclude 1 in intestine, then exclude 2 in kidney. So glued 1 in RBC, kidney and brain, glued 2 in serosal side of intestinal cells, liver, then pancreas is also there. Then glued 3 brain neurons, glued 4 heart, skeletal muscle adipose, glued 5 small intestine, testis and kidney, glued 7 liver, endoplasmic reticulum, then exclude 1 intestine, exclude 2 kidney. So this is all about the first chapter. So hope you understand something. And കുറേ കാര്യങ്ങൾ ഇതിൽ എക്സ്പ്ലെയിൻ ചെയ്യാത്തതുണ്ട്. അത് ഏറ്റവും ഇമ്പോർട്ടൻ്റ് ആയിട്ട് ചോദിച്ചത് മാത്രമേ ഇതിലിപ്പം ഡിസ്കസ് ചെയ്തിട്ടുള്ളൂ. അപ്പം സോ താങ്ക് യു.